There is something living inside your DNA that scientists cannot yet find in any fossil record, cannot assign to any skull in any museum, cannot point to in any archaeological dig site anywhere on the planet. It predates the woolly mammoth. It predates Neanderthals. It predates our entire genus, as it's currently understood by mainstream paleoanthropology. And yet the evidence for its existence is sitting right now in the genomes of billions of living people, measurable, reproducible, published in peer-reviewed journals by researchers at some of the most rigorous scientific institutions in the world. Scientists have been calling it a ghost population. And in March 2025, a team from the University of Cambridge published findings that forced the field to confront just how significant that ghost actually is, and how badly the standard story of human origins needed to be revised. To understand what the Cambridge team found, you have to understand what the term ghost population means in genetics, because it's a specific technical concept rather than a vague metaphor. In evolutionary genetics, a ghost population is defined as an ancestral group whose existence can be inferred from the genetic signatures it left in the DNA of living descendants, but for which no direct physical evidence in the form of bones, teeth, or sequenced ancient DNA has yet been found. The ghost is detectable not because we've dug up its remains, but because the people alive today carry a proportion of DNA that statistically cannot be explained without invoking an ancient interbreeding event with something distinct from the populations we already know about. The ghost shows up as an anomaly in the genome, a cluster of genetic variants too old, too divergent, and too widespread to have originated within the lineages we've identified, pointing backward in time like an arrow towards something that lived and died and left pieces of itself in us, before anyone knew it existed. The Cambridge study, published in Nature Genetics in March 2025 and led by a team including geneticist Aylwin Scally, introduced a new computational modeling tool called COBRA, which stands for Coalescence-Based Reconstruction and Analysis of Ancestral Structure. Unlike earlier statistical approaches that analyzed specific portions of the genome looking for unusually archaic sequences, COBRA works across full genome sequences, building a probabilistic model of how ancestral populations could have split, evolved independently, and then rejoined at different points in time, then testing which model best fits the actual pattern of genetic variation observed in thousands of modern human genomes drawn from the 1000 Genomes Project, one of the most comprehensive databases of modern human genetic diversity assembled to date. What COBRA found when applied to this dataset was not what the researchers expected to confirm. The model that best fits the actual observed pattern of human genetic variation is not a single ancestral lineage that gradually diverged into modern humans. It's a much more complex scenario. Approximately 1. Populations remained separate for over 1 million years, evolving independently in isolation. Then, approximately 300,000 years ago, they came back together in a major admixture event, merging into a single ancestral pool from which all modern humans descend. The larger population contributed roughly 80% of the genetic material that exists in modern human genomes. The smaller, previously undescribed ghost population contributed the remaining 20%. Those numbers deserve a moment to be properly understood, because the 20% figure is not a trivial genetic footnote. For comparison, all the Neanderthal DNA in a modern European or Asian person amounts to between 1 and 4% of their genome. All the Denisovan DNA in a modern Melanesian person amounts to 4 to 6%. The ghost population identified by the Cambridge study contributed roughly 5 times more genetic material to modern humans than Neanderthals did, and roughly 3 to 4 times more than Denisovans did in the populations most heavily influenced by Denisovan interbreeding. This is not a minor signal at the edge of detection. It's a major structural feature of the modern human genome that previous models were too limited to see clearly, and that COBRA has now demonstrated is present consistently in all modern humans, regardless of their geographic origin or ethnic background. That last point is one of the most striking aspects of the finding. Denisovan contribution is concentrated in populations with ancestry from Eastern and Southeast Asia and Oceania, but the ghost population signal found by the Cambridge team isn't geographically restricted in the same way. It appears to be a shared feature of all modern human ancestry, not a local admixture event that affected some populations and not others, but a deep, ancient mixing that happened before the divergence of any of the modern human population groups that currently exist. Co-author Aylwin Scally described the finding in public statements following publication as evidence that human history is far richer and more complex than previously imagined, and that the ability to reconstruct events from hundreds of thousands or millions of years ago just by analyzing DNA from living people remains, in his words, astonishing. The identity of this ghost population is currently unknown in the most literal sense. There are no candidate fossils, no skeletal remains, no directly sequenced ancient genomes that researchers have been able to match to the ghost signal. At 1. forms whose exact evolutionary relationships remain debated. What the COBRA analysis can establish is the timing of the divergence and the proportion of contribution, but not the physical form or specific identity of the ghost population. Its fossils may simply not have been found yet. Preservation of hominin remains from one. 5 million years ago, particularly in sub-Saharan Africa, where ancient DNA recovery is nearly impossible due to heat and humidity destroying genetic material in bones within decades rather than millennia, is extraordinarily sparse. The ghost may have left bones in the ground somewhere that no one has excavated. It may have left bones that have already been found, but haven't yet been correctly identified as belonging to the lineage in question. Or it may simply be unidentifiable from any available physical evidence, known only, and perhaps permanently, through what it wrote into the genomes of the people alive today. What makes the Cambridge ghost finding particularly significant is that it sits within a broader, rapidly building picture of archaic ghost populations contributing to modern human genomes in multiple geographically distinct contexts. A picture that has been assembling piece by piece over the past decade, and that now, taken together, looks nothing like the simple, clean origin story that dominated paleoanthropology for most of the 20th century. The first well-documented ghost population signal outside the Neanderthal and Denisovan discoveries came from West Africa. In a study published around 2020, researchers comparing whole genome data from modern Yoruba and Menda populations in West Africa with the genomes of Neanderthals and Denisovans found a portion of genetic material that didn't match any known archaic human genome, deriving between 2 and 19% of their ancestry from a lineage that had never been identified from any physical fossil. This West African ghost population appears to have diverged from the main human lineage even earlier than Neanderthals, making it, if confirmed, one of the most deeply divergent archaic human groups ever identified, and it appears to have interacted and interbred with the ancestors of modern West Africans at some point during the period when modern humans were establishing themselves across the African continent. Because ancient DNA preservation in West Africa is essentially impossible, with current technology, due to the hot, humid climate, rapidly destroying genetic material in any biological remains, the only evidence for this ghost population's existence is the signal it left in living people. A separate ghost population signal has been detected in the genomes of indigenous populations from Southeast Asia and the Pacific, distinct from the Denisovan signal and pointing to yet another archaic group that interbred with modern humans as they moved through the region. A 2019 study identified what appeared to be traces of a previously unknown hominin exclusively in the population of Flores, Indonesia, the same island where Homo floresiensis, the small-brained archaic human nicknamed the Hobbit, was discovered from fossil remains, raising the question of whether this island ghost signal might represent genetic traces of exactly that lineage, though the connection remains unconfirmed. Alan Rogers, a population geneticist at the University of Utah, has published research identifying what he calls super-archaic populations, groups that diverged from the mainstream of human evolution even earlier, between 2 and 1. 8 million years ago by his estimates, and that appear to have interbred with archaic human lineages, including the ancestors of Denisovans, leaving traces that survive today in genomes of modern people through this chain of introgression events. These super-archaic populations would be so ancient that their divergence predates not just the emergence of Homo sapiens, but potentially the emergence of the Homo janus in Africa entirely, pointing toward lineages even more deeply separated from modern human ancestry than the Cambridge ghost, and raising genuinely profound questions about the complexity of early human evolution in Africa over the past 2 million years. What has been emerging most clearly from these various findings, taken together, is that interbreeding between distinct hominin groups was not an exceptional rare occurrence in human prehistory. It was apparently routine, repeated, and geographically widespread. Every time modern humans or their archaic relatives moved into a new area or came into contact with a previously isolated group, interbreeding appears to have followed as a matter of course, leaving genetic traces that modern population genomics tools are only now sophisticated enough to detect and quantify with confidence. This has a straightforward biological explanation. Hominin groups that had been separated for tens or hundreds of thousands of years were different enough genetically to be detectable as distinct in modern genomes, but similar enough biologically to produce fertile offspring without significant reproductive barriers, meaning natural selection could and did act on the novel genetic combinations those offspring carried, preserving beneficial variants from archaic groups in the genomes of their descendants. Some of those preserved variants turn out to matter biologically in ways that directly connect ancient admixture to modern human traits and capabilities. The Cambridge team noted that genes from the ghost population appeared to be particularly concentrated in regions of the genome associated with brain development and cognitive function, raising the possibility that the 20% contribution from this unknown archaic lineage may have included genetic variants that shaped the cognitive evolution of modern humans in significant ways. This is preliminary and requires further research, but the pattern is consistent with what has been found in other documented archaic introgression events. The EPAS1 altitude adaptation gene inherited by Tibetans from Denisovans is one well-established example. Modern immune system genes, including several variants in the HLA system that controls how the body identifies and fights pathogens, show strong signatures of archaic introgression, with variants inherited from Neanderthals and Denisovans apparently providing advantageous responses to local pathogen environments that modern humans encountered as they spread into new regions. The ghost population's contributions to brain-related genes, if confirmed through further analysis, would extend this pattern of archaic introgression, contributing functionally important biological variation to a domain of human biology that has long been considered distinctly modern. The technical tool at the heart of the Cambridge study, COBRA, is worth understanding as a development in its own right, because the kind of computational approach it represents is increasingly what's driving new discoveries in this field. As the database of sequenced modern human genomes has grown from hundreds to thousands to tens of thousands of full sequences over the past decade, the statistical power available to detect ancient population structure has grown with it. Earlier methods for detecting ghost populations had to make simplifying assumptions about ancestral population sizes and interbreeding timing, assumptions that limited their sensitivity to relatively recent events or large admixture proportions. COBRA-A was designed specifically to relax these assumptions, explicitly modeling ancestral population splits and rejoins across a broader parameter space, and testing which model configurations best explain the actual observed variation patterns in thousands of modern genomes simultaneously. The fact that it identified a ghost population contributing 20% of modern human ancestry that earlier methods had either Every major ancient DNA discovery of the past 15 years has added complexity to this picture, and the 2025 Cambridge study adds the most structural revision yet, suggesting that even the African origin story itself is not a single lineage emergence, but rather a merger of two deeply separated populations, one of them previously unknown to science, occurring roughly 300,000 years ago. The modern human who left Africa to populate the rest of the world was already, before taking a single step out of the continent, a genetic hybrid carrying the legacy of at least two ancient lineages separated by over 1 million years of independent evolution. What remains genuinely, honestly unknown is how many more ghosts are still hidden in human genomes waiting to be found. Each methodological advance in population genetics has revealed population structure and archaic admixture that previous methods couldn't detect, and there's no compelling scientific reason to think COBRA has identified the complete picture rather than added one more layer to a story that may be far more complex still. The researchers themselves have been careful to note that their model fits the data well, but doesn't exclude more complex scenarios involving additional archaic populations or multiple admixture events. As tools improve further and the database of both modern and ancient human genomes continues to grow, it's entirely plausible that additional ghost signals will be identified, each one representing a people who lived and evolved and eventually disappeared as a distinct group, leaving no fossil record anyone has found. No tool assemblage anyone has attributed to them, no cave art anyone has seen, but surviving in pieces inside every person alive today, whispering through the genome, a history nobody knew was there. There's a specific and underappreciated dimension of the ghost population story that almost never makes it into popular coverage, and it concerns what these findings mean, not just for our understanding of human prehistory, but for our understanding of human biology right now in living bodies, expressed in traits and vulnerabilities and capabilities that are directly traceable to these ancient mixing events if you know where to look. The standard framing of archaic introgression in popular science coverage tends toward the exotic, the idea that carrying Neanderthal DNA is a curious historical footnote, an interesting fact about human origins with no particular present-day significance. The actual genetic evidence tells a more immediately relevant story. Several of the most important immune system variants in modern human populations trace directly to archaic introgression events. Variants in the HLA system, the set of genes that controls how the immune system identifies foreign pathogens and distinguishes them from the body's own cells, show among the strongest signals of archaic introgression anywhere in the modern human genome. Specific HLA variants inherited from Neanderthals and Denisovans are found at high frequency in modern populations from regions where those archaic groups previously lived. And the most compelling explanation for why these variants spread and persisted, rather than being diluted out over generations, is that they conferred genuine survival advantages, providing immune responses to pathogens that early modern humans encountered as they moved into new environments that Neanderthals and Denisovans had been adapting to for hundreds of thousands of years before our ancestors arrived. In effect, interbreeding with archaic humans functioned partly as a biological shortcut, allowing modern humans to rapidly acquire locally optimized immune configurations that would otherwise have required tens of thousands of years of independent natural selection to evolve from scratch. The same logic applies to the ghost population's contribution, which the Cambridge team has noted appears concentrated in brain-related genomic regions. If variants from the ghost population affecting neural development and cognitive function were genuinely retained and spread in modern human populations, the most parsimonious explanation, following exactly the same reasoning as the immune gene case, is that those variants were advantageous, that they contributed something to human cognitive capability that was worth keeping. This is speculative and requires considerable further research before any specific functional claim can be confidently made, and the researchers involved have been appropriately careful about how strongly they frame this particular finding. But the structural pattern is worth taking seriously, because it suggests the possibility that some aspects of distinctively modern human cognition, whatever that ultimately means in neuroscientific terms, may not have evolved entirely within a single unbroken lineage, but may represent a mosaic assembled partly from contributions of archaic groups whose specific identities we haven't yet been able to recover from any physical fossil. There's also a genuinely important methodological story embedded in how these ghost population discoveries are being made, one that connects the ghost population research to broader developments in computational biology that are reshaping multiple fields simultaneously. The population genomics tools being used to detect these ancient signals, COBRA in the Cambridge study, similar Bayesian inference frameworks in other research groups, are fundamentally statistical tools for solving an inverse problem. You observe the current state of something, modern human genome sequences, and you work backward computationally, modeling what historical processes could plausibly have produced that observed pattern. This inverse approach is powerful because it doesn't require you to have direct evidence of the historical events in question. You don't need a fossil from 5 million years ago to prove the ghost population existed. You need a sufficiently large, well-characterized database of modern genomes and a computational model that can distinguish between different possible historical scenarios with enough statistical confidence to make a meaningful claim. What's changed dramatically in the past decade is both sides of this equation simultaneously. The database of sequenced modern human genomes has grown from a few hundred to tens of thousands and continues growing, providing a far richer and more globally representative picture of modern human genetic variation than existed even 5 years ago. And the computational models have become far more sophisticated, moving from relatively simple two-population models toward the kind of multi-population, multi-event modeling that COBRA represents, capable of detecting older, smaller, and more complex admixture signals that earlier methods would have either missed entirely or misattributed to other causes. This convergence of more data and better tools is what's producing the acceleration of ghost population discoveries we've seen over the past several years. And there's no particular reason to think the rate of discovery will slow down rather than accelerate further as sequencing costs continue to fall and genome databases continue to expand. It's worth being explicit about the relationship between these genomic ghost population findings and the ancient DNA field more broadly, because they represent two different but complementary approaches to the same underlying historical question. Ancient DNA research, the extraction and sequencing of genetic material from actual fossil, remains like the mammoth teeth and Denisovan finger bone discussed earlier, provides direct evidence of specific populations at specific times and places, but is limited by the availability of preserved biological material, which is heavily biased toward cold, dry environments and relatively recent time periods. Population genomics, the statistical analysis of modern genome sequences to infer ancestral history, can potentially reach much further back in time and doesn't require any fossil preservation at all, but provides indirect, statistically inferred evidence rather than direct sequencing of the archaic genomes themselves. The two approaches ideally complement each other, with ancient DNA providing ground truth validation of population genomic inferences wherever preserved material exists, and population genomics extending the picture into time periods and geographic regions where ancient DNA recovery is impossible. The ghost population identified by the Cambridge Cobra analysis sits in a time period and likely a geographic context where ancient DNA recovery from hominin remains is currently impossible. Temperatures in sub-Saharan Africa rapidly destroy DNA in biological remains, meaning the gap in the fossil record created by poor DNA preservation in the African context is likely permanent rather than a temporary limitation of current technology. This makes the population genomic approach, reading the ghost's presence from signals it left in living people, not just the currently available method, but potentially the only method that will ever be available for understanding this specific archaic lineage in genetic terms, regardless of how much the technology improves. If its fossils exist in African soil, they may eventually be found and recognized morphologically, but the sequencing of their genomes may remain permanently out of reach, making the signal in living human genomes the primary, and possibly sole, source of genetic information about who they were and what they contributed. There's a broader philosophical point worth drawing out from all of this, one that the researchers themselves have touched on in interviews following the Cambridge publication. The story that human beings have been telling about their own origins for the past century, the story of a single triumphant species emerging in Africa and spreading out to replace or displace everything that came before, was never really supported by the fossil evidence as firmly as it sometimes appeared. The fossil record of human evolution has always been messier, more fragmentary, and more ambiguous than textbook simplification suggested. What ancient DNA and population genomics have done is reveal that this messiness reflects real biological complexity rather than gaps in the record, that human evolutionary history genuinely was a tangled, repeated process of divergence and reunion rather than a clean linear progression, and that the people alive today are the result of that tangling, carrying in their genomes the accumulated genetic legacy of multiple ancient lineages, some named and documented, and at least one that existed, flourished for over 1 million years, and contributed 1/5 of the modern human genome, before disappearing from the physical record so thoroughly that it left no trace science could find for all the years of paleoanthropology that preceded the genomic revolution. That ghost was always there. It just took the right tools, applied to enough data to finally see it.